Hello everyone, welcome to the world of biology. We are in the chapter number 6 and today we will start part number 5. Although its theory I have already explained the type of genes. As far as type of genes are concerned, on the basis of transcription, it may be monocystonic or polycystonic. Monocystonic genes are found in eukaryotes while polycystonic genes are found in prokaryotes. What do you mean by the eukaryotic monocystonic gene? What will you see? It is a continuous sequence of the polynucleotides that codes a single protein. So you can see it will form a single protein or a single enzyme, that is the monocystonic gene. On the other hand, in the prokaryotes, it has polycystonic gene that is made of different types of nucleotide chains where a part will form protein number first, another will form protein number second, and the next will form protein number third. It means polycystonic genes codes for many proteins at a time, while on the other hand in monocystonic gene, one gene will code a single protein at a time. So this is about the type of genes. Now we will start the next topic, that is transcription is here, and the process of transcription. As far as transcription is concerned, the formation of RNA from DNA is known as transcription. This process is carried out in three important steps. First of all, chain initiation step. The second one is elongation step. And the third one is termination step. So what will you see? The transcription begins with initiation and it requires a specific enzyme, that is RNA polymerase enzyme. And basically this enzyme is DNA dependent. So we will discuss about the process of transcription. For more details, come on to the PPTs. So in the monocystonic genes, you can see this is eukaryotic mRNA is given, and this is red colored structure will code the protein, and a single protein is coded here. This is the capping at 5' position and tailing at 3' position. On the other hand, in the polycystonic genes, you can see it has different translational sites. Here protein 1 is coded, protein 2 is coded, protein 3 is coded. Now come to the next topic, that is process of transcription in prokaryotes. There is a single DNA dependent RNA polymerase that catalyzes transcription or synthesis of all three types of RNAs in a bacteria. So what will you see in prokaryotic cells, or you can see in the bacteria, there will be a only single type of RNA polymerase that will catalyze all type of RNAs. The process of transcription is completed in three steps. The first is initiation, elongation and termination. In the very first point, initiation of transcription. So RNA polymerase binds to promoter. Promoter gene is present where the RNA polymerase enzyme will bind and initiate the transcription by associating transiently with initiation factor sigma. So sigma factor will initiate the transcription. It uses NTPs as substrate and polymerizes in template dependent fashion obeying the rule of complementarity. It also facilitates opening of the helix and continuous elongation. RNA polymerase does not require a primer to initiate transcription. As we have already seen in the replication, the process requires the RNA primer there, but here primer is absent. Elongation of the chain, RNA polymerase unzip the DNA double helix and form an open loop. So what will you see? RNA polymerase will unzip the DNA double helix, will form an loop, and it uses NTPs as substrate and polymerizes in a DNA template following the rule of complementarity. Only a short stretch of polymerized RNA remains bind with the enzyme, and the process of polymerization continued till the enzyme reaches the terminator gene. In the termination of the gene, you can see once the polymerase reaches the terminator region of DNA, which is GC rich region. GC rich means it contains guanine and cytosine and has a hairpin like structure. The nascent RNA fall off. So also the RNA polymerase. So RNA and RNA polymerase will be removed from there. This results in termination of transcription. The RNA polymerase is only capable to catalyzing the process of elongation. It associates transiently with initiation factor sigma and termination factor rho to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. RNA polymerase recognizes the terminator gene by a termination factor called rho factor. So rho factor is the termination factor. You can see the figure 6.10 of your book. This is the DNA double helix, and this orange colored round structure is showing the RNA polymerase. As sigma factor will bind to RNA polymerase, then it will start the initiation. As the RNA polymerization continues, or you can say RNA formation continues, it will unzip and continue its process, and sigma factor will come together till the terminator gene is achieved. This is the elongation process, and in the finally sigma factor is removed and rho factor dominant over the RNA polymerase, and it will remove the RNA polymerase and it will stop the transcription. Special note: in prokaryotes, bacteria does not require any processing to become active because the bacterial proteins or enzymes are very small in size and they need not any processing. Both transcription and translation takes place in the same compartment because there is no separation of nucleus and cytosol. This is the another one most important point. In the bacterial cell, there is no separation in nucleus and cytosol, or you can say cytoplasm. So both the processes carried out in same place. The translation can't start much before the translation can start much before the mRNA is fully transcribed because it is completed parallel to each other. Both transcription and translation can be coupled. Coupled means both the process carried out at the same time. Question and intriguing question is that how is the RNA polymerase able to catalyze the all three steps, which are initiation, elongation and termination. Now the next one is additional complexities in eukaryotes. Eukaryotes has much more complexities. There are at least 3 different types of RNA polymerase enzymes in the nucleus. There are 3 types of RNA polymerase enzymes are found. In addition to the RNA polymerase found in the organelles. Cell organelles also possess the RNA polymerase. That is different from that. It shows the clear-cut division of labor. So the presence of different type of RNA polymerase is the symbol of division of labor. What are the roles of the different RNA polymerase enzymes? You can see RNA polymerase first, RNA polymerase second, RNA polymerase third. RNA polymerase first transcribes the rRNA, and there are different types of rRNAs in the eukaryotes: 28S rRNA, 18S rRNA, 5.8S rRNA. On the other hand, RNA polymerase second transcribes the precursor of mRNA, the heterogeneous nuclear RNA. It is also known as hnRNA. While RNA polymerase third transcribes the tRNA, 5S rRNA and snRNA. So you can see the division of label. A table is given here. These are the forms of RNAs: RNA pol first, second, third. These are the products and location. RNA polymerase found in the nucleolus, while rest of the two other RNA polymerase enzymes found in nucleoplasm. Come to the next. In the eukaryotes, the primary transcript contains both the exons and introns. You can see exons are the functional part of the RNA, while introns are the non-functional part of the RNA. So it requires a process called post-transcriptional processing that occurred in the nucleus. So important point is that RNA processing is completed in the nucleus. It is completed in three steps. The first is capping at the 5' end. At the 5' end, 5,7G nucleotide will attach. On the other hand, at the 3' end, addition of poly-A tail, AAA, adenosine will bind to the 3' end. It will form a long poly-A tail, and the splicing to remove introns. What will you see? Introns are removed from the hnRNA. You can see the steps. What is capping? Capping at 5' end, an unusual nucleotide called 7-methyl guanosine triphosphate (MZTP) is added to the 5' end of the hnRNA. The cap protects the 5' end of the mRNA from degradation by nuclease enzymes, and also helps the position the mRNA correctly on the ribosome during protein synthesis. So it plays two important roles. First of all, it will protect from the degradation, and second, it will help to position on mRNA correctly. What is tailing? Tailing at the 3' end. In this process, adenylate residues, almost 200 to 300 adenylate residues are added at 3' and in a template dependent manner. There is evidence that the poly-A tail plays a role in efficient translation of mRNA as well as in the stability of mRNA. So also it plays two important roles: efficient translation and the stability of mRNA. And the final one is splicing. Splicing means to remove the introns and exons are joined together in a proper order to allow the translation. So it requires a specific enzyme called spliceosome. Spliceosome is responsible for splicing in the RNA, hnRNA. Spliceosome is made up of proteins and small RNAs that are associated to form proteins RNA enzymes called smaller nuclear ribonucleoproteins or RNPs, pronounced as SNURPs. The cap and the poly-A tail on a mRNA are also indications that the mRNA is complete. The presence of cap and tail is the symbol of complete mRNA, and it is called not defective. You can say non-defective. And the processed hnRNA is now allowed mRNA and transported out the nucleus for translation. So you can see this is the intron, and these snRNPs will cut this part and rest of the part comes closer to each other. You can see the process of transcription in eukaryotes. This is the DNA. You can see these yellow and orange strips in the RNA, that is known as the hnRNA, where yellow colored structure is exon, or you can say useful part of the RNA. Introns are the useless part of the RNA. At the 5' position, an unusual Methylated guanosine triphosphate will attach, and on the other hand, in the 3' position, polyadenylation will complete and polyatal is formed. And what will you see? These rounded structures are showing the spliceosomes that are cutting the orange colored part, and now it becomes active mRNA, and this is known as process of transcription in eukaryotes. Now, next point, genetic code. What is genetic code? The perfect definition is given here. The genetic code is the set of rules used by living cells to translate information encoded within genetic material into proteins. So you can see genetic code is a triplet codon codes which is responsible for the translation of proteins or amino acids. The process of replication and transcription based on complementarity. Similarly, it will follow the complementarity. The process of translation is the transfer of genetic information from a polymer to nucleotide to a polymer of amino acids. There is no complementarity exists between nucleotides and amino acids. This is important. If there is change in the nucleic acid genetic material, there a change in the amino acid in proteins is also seen. So there must be a genetic code that could direct the sequence of amino acids in the protein during translation. So in the discovery of genetic code, it required the involvement of different streams of scientists like physicists, chemists, biochemists, and geneticists. George Gamow, 1953. He was from the physics area. He was a physicist. Proposed that the code should be combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. So George Gamow was the first person who told that the genetic codes are made of 3 nucleotides. It was a very bold proposition because a permutation combination of 4 to the power 3, 4 multiplied by 4 multiplied by 4 would generate 64 codons, generating many more codons than required. On the other hand, there were another scientist, H.G. Khorana. He was from India. He born in India and worked in America, United States of America. So H.G. Khorana enables instrumental synthesizing RNA molecule with desired combinations of bases, homopolymer and copolymers. There is another one more scientist, Marshall Nirenberg's cell. Cell-free system for protein synthesis finally helped the code to be sorry deciphered the discovery of genetic codes. There is one more scientist, Severo Okao, was also helpful in polymerizing RNA with desired sequence in a template-independent manner, enzymatic synthesis of RNA. And the enzyme was named on him. And finally, checkerboard was prepared by Marshall Nirenberg, in which 64 codons were established. Suppose type of code and number of permutations. Suppose if the genetic code was made of single nucleotide, then there will be only 4 number of nucleotides. Genetic code is doublet, then number of codon will be 64, and triplet 64, 16 and 64. So you can say 64 codons are sufficient, and these are the wrong combinations. Only the triplet code really looks feasible. The salient features of genetic code. What are the important features of the genetic code? The first is the codon is triplet. It is made of 3 nucleotide sequences, and 3 nitrogen base sequences constitute 1 codon. There are 64 codons, out of which 61 codes for amino acids and 3 codons are stop codons, also known as nonsense codons. They are the useless, they will stop the transcription. Stop codons do not code for amino acid and has no anticodon on the tRNA. Example, the 3 stop codons have been named as follows: amber, opal and ochre. You can see look at the spell AMBER. This is UAZOPL, or the another name of this is UMBER. That is UZA, and OCHRE, ochre, that is UAA. So it will help in the termination of the transcription. One codon codes for only one amino acid, hence it is unambiguous and specific. Degeneracy, what do you mean by the degeneracy? Some amino acids are coded by more than one codons. You can see serine, it is coded by 6 different type of codons. The codon is real in mRNA, the codon is read in mRNA in a contiguous fashion. There are no punctuations, means comma and gap, etc. Codons are universal, for example from bacteria to human, UUU codes for phenylalanine, but there are some exceptions. You can see some exceptions to this rule have been found in mitochondrial codons and the some protozoa. AUG has the dual function. It has the two function. It codes for methionine and it also acts as initiator codon. Non-overlapping, the genetic codes reads linearly, not the overlaps, and direction, the code only read in 5' to 3' direction. An anticodon, each codon has a complementary anticodon on the tRNA. So in the following is the sequence of nucleotides in mRNA. Predict the sequence of amino acids coded by it. Take the help of checkerboard. So suppose this is the mRNA. A U G U U U U U U C U U C U U C U U U U U U U U C. Then this it will code for methionine, phenylalanine, aniline, and so on. Now try the opposite. Following is the sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. Methionine, phenylalanine. So there will be some sort of difficult, because one amino acid can be coded by more than one codons. So it is difficult to find out the exact sequence of codons in mRNA. This is a table taken from your book. It is made of 64 codons. Out of that you can see UAA, UAG and UGA. These are the stop codons, while AUG, it is coding the methionine, it is initiation codon. Now come to the next point, mutation. Although in the chapter number 5 we have discussed about the mutation, but have a look again in a brief. Mutation is a phenomena which results in sudden alteration of DNA sequence and consequently results in changes in the genotype and phenotypes of an organism. It is first seen by Cartwright in Anconship and it is discovered by Hugo de Vries. I have already explained all these things in chapter number 5. Mutation and genetic code. Relationship between DNA and genes are best understood by mutation. Regions of the mutation already explained: deletion, duplication, inversion, translocation. Point mutation, it is a small scale mutation characterized and is arose due to the change in a single base pair of DNA. This is known as point mutation. It changes the base sequence and causes frame shift mutation. It is caused due to deletion, insertion or duplication. You can see the example of point mutation is sickle cell anemia. What is sickle cell anemia? Sickle cell anemia is an inherited red blood cell disorder in which there aren't enough healthy red blood cells to carry oxygen throughout your body. Regions of sickle cell anemia: the defect sorry is caused due to substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. So substitution of amino acid takes place due to single base substitution at the 6th codon of the beta globin gene GAG to GUG. So A is replaced by U. Frame shift mutation, it occurs due to frame shift, insertion of frame shift, deletion of the one or the more in nitrogen bases in the gene. There is a change in the whole sequence of amino acid from the point of insertion or the deletion, that best understood in beta thalassemia. You can see the frame shift mutation can easily be understood by following example. Consider a statement made up of following words, each having three letters like genetic code. Ram has red cap. You can see each word is made of three alphabets. Now insert B in between has and red. So what you need to do, these red colored alphabets are inserted after the sequence. Ram has BREDCA. Again you will form combination of three alphabets, but P is now separated. When you will add I in between the B, after B then BIR, EDC and AP, and when you have added G, then what will you see? Now insert I after B, now insert G after I, then it shows insertion. So Ram has big red cap. It means this sequence have changed the sentence. Similarly, it can be repeated by deleting the letters like RED one by one. Then what will you see? Ram has red cap, Ram has EDCAP, Ram has DCAP, and finally Ram has cap. So what is the conclusion? The above exercise shows the insertion deletion of half bases changes the reading from frame from the point of insertion or of three or its multiple bases, insert or delete or one multiple codons. You can see the effect of point mutation. Suppose TTC, this is the sequence in DNA AAG, it will form the lysine. So what happened here, C is replaced by T. Then in the mRNA there will be the A, and if it is coding the same type of amino acid, then this is known as silent mutation. Here is no mutation, this is silent mutation. And suppose in the TTC initial T is replaced by A, then it becomes ATC, it will code the UAG, that is the stop codon. It is known as nonsense codon. Nonsense means which stop the transcription, and missense means when it will form other than the lysine. So dear students, today we will stay here. Try to understand all the aspects. If you have any problem, you can message me. Thank you very much. Have a nice day.